tonight is going to build a good bit on what we did last week. So those who weren't here, I hope you got a chance to maybe Zoom it because, yeah. Assuming that you either have this from last week or you picked up one now, or at least you remember, because I'm going to be referring to this throughout, presuming that you remember if you were here, because that will serve as a kind of a backdrop. Okay? Oh, come on over here and pick up papers. Here last time, you know the deal. And let's see, these you should have already got. These are for next week. Okay, so this evening we're supposed to be thinking about the origin of life from a scientific perspective, that is. Okay. Now, if you take out your God, man, word, world diagram, just to be reminded that that lower right-hand corner where we've got us at the bottom, man looking up to word and world, but now looking up to the world, and the arrow that we had drawn there representing the scientific process, that is, we humans looking at the physical realm, trying to figure it out, and among other ways, like the arts is the other easy example, science is a way to do that. This diagram is very helpful to help us do that. And so I'm bringing that up again, and also for folks that weren't here last time, to remind you of the point that one of the values of this diagram is to allow us to look at God's world, God's word, without, I hope, getting all hung up with, oh my goodness, I'm a Christian, I can't think about science, or whatever way you've found this to be a challenge. And I used this approach with my students at Taylor for many years, hoping that, among other things, they would leave no longer fearing science, no longer being ashamed to admit that, well, yeah, I majored in biology or chemistry because I want to be a doctor. So the excuse for taking science was to be a doctor, which seemed biblical enough for most. But to get all of my students, regardless of what their perceived career path might be, to not have to kind of cover up the fact that you're a Christian who likes science. And so that diagram puts that activity of looking at the physical realm as a scientist, that is, looking for physical answers to physical questions, because that will apply this evening and next week as well. So just to remind you of the utility of this model. Now, as I was rereading the papers for tonight, and I was quite pleased when I went looking, because I knew I was supposed to do something about the origin of life, and I keep up with the literature, but not like I did when I was actually doing science, and not like when I was teaching, but I still try to keep up with not the primary professional literature, but sort of the secondary literature. And two examples of that I get. One is called The Scientist, which in spite of that generic title actually is largely limited to life sciences. And this is American. Comes out quarterly, more or less. And one of the papers is from this guy. And then The New Scientist, which is a weekly from Britain. And both of these are aimed at an audience that, if not people in careers that are somehow tied to biology, at least to folks that find things interesting. And this is a way to keep up. And I was quite pleased as I was pondering, well, what am I going to use to talk about the origin of life, that in these two issues that are both December 2025 and winter 2025. Like, great, because, rather than, well, I like to speak from known sources of known repute. So these are both perfectly legitimate to allow us to enter the world of scientists with respect to these two life sciences. So if you take a look at those two handouts, both of which are kind of colorful, you'll notice on the first page of one, and maybe both, one anyway, you can see the name of the publication, you can see the date. And that was one of the things that I really tried to get my students to pay attention. I mean, I've said this last week, but again, that when you're working in any field and you're utilizing sources, it really is your responsibility to, as best as you can, understand the nature of those sources. And so what I've just told you for the papers that you read for this evening are that they come from very recent, scientifically legitimate sources. Same thing as when you're studying scripture and you're using commentaries, and whether the commentary is put out by the Baptists or the Presbyterians or academics, you know, you're going to get a very different take on scripture. So it helps in any area to know what your source is, the nature of it as best you can. And as I was reviewing those papers, found myself thinking, okay, I'm comfortable with a lot of the terminology and so forth, and I can't assume that for the audience. And I even said to Yu Yang, my wife, earlier today, you know, I don't know where I should start. Decided I better start at the very beginning. Yeah, here we go again. Okay, which is to say some basic drum roll, drones, chemistry. Yay, chemistry. Okay, and the physicist in the crowd spotted water. Let's go to the board and do a few simple things here. Okay, this is a circle. That's pretty good. Well, pretty crummy. It also can be used as the representation for oxygen, or it could, in this case, be the representation of one atom of oxygen, where atoms are the smallest. T-shirt?
Atoms make up everything, don't trust them.
Yeah. Atoms make up everything, don't trust them. Speaking again in case the Zoom folks didn't quite hear it. Okay. So that, without going into any subatomic detail, is supposed to be an oxygen atom. Likewise, hydrogen. I'll put two of them up here, imagine that.
It should be a different color.
Hey, you want to come over and do this? Okay. I actually did. I used to throw things at my students, but I wouldn't look. I would just.
Pretty obvious that it's hydrogen if it's red.
What kind of obvious is that?
Intuitively obvious to even the most casual observer.
We have lots of. We've known each other since 1981, so you know. I'm gonna. No, I'm just gonna use the chisel edge so that we've got a more visible representation. Now, what color would you prefer? Is black okay?
Yes.
It's okay. Once you got the red, now you can use black.
Okay, so now we're gonna do two oxygen, or two, well, here we go. Hydrogen, 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 hydrogen. And I could draw some lines in here and here, indicating that now these two hydrogens are chemically linked to this oxygen. Likewise over here, when you have substances, that is physical things that are they're bigger than atoms. So a few atoms stuck to each other, those are molecules. Molecule of water, molecule of water. Everybody okay so far? Not too painful? Okay. Too simple? All right, moving on. In the readings, you saw the phrase organic this, organic that. And organic is used in a lot of ways that I'm not entirely comfortable with, but the way I am comfortable with it is in terms of organic chemistry. Okay. And organic chemistry means the chemistry of substances whose molecules have the carbon bonds. So, oh dear, what color was, did we do carbon yet? We're gonna do black for carbon because you know, graphite and cobalt stuff. Okay. There's the carbon. Now, what do we do hydrogen? Remember?
Right.
Red. Okay. I don't remember. Some color. Okay. 1, 2, 3, 4. Hydrogen is stuck to a carbon. What's that stuff?
Nothing.
Methane. Okay, pseudo trick question. Is that an organic molecule? It's natural gas. There are no carbon to carbon bonds, and yet because it gets lumped in because all you gotta do, you know. Just do that. Finish that. And now we got an organic molecule because there is a carbon to carbon bond. So in your reading for tonight, that's how the word organic is being used to refer to substances, more than one atom stuck together, substances that contain carbon to carbon bonds. So the cool thing about carbon, among other things, is if you make typically 4 covalent linkages, covalent bonds, 2, 3, 4, 1, 2, 4, 5, 6, 7, 8. Okay. This can go on semi-indefinitely. Organic molecules can be that short, or they can be quite long. And I won't put in all the bonds. I'm just going to move this right along here. Anybody know what I'm drawing here? Clearly, it's an organic molecule because look at all the carbon to carbon bonds.
It's like a gold post.
Octane, a major component gasoline. There it is. Now, you look at, well, DNA is across here, but it's got carbon and a bunch of other stuff. But carbon-to-carbon bonds is part of its central structure, and I forget, do I have a lipid on here someplace? Yeah, you can't, it's too big. Anyway, that's a phospholipid, which is one of the major components of cell membranes. I can go on and on, but I won't. My point is, though, I've now introduced you to what the organic word is being used to describe and its importance in the kind of chemistry that living things require. The carbon to carbon to carbon to carbon allows building all kinds of molecules because now you can dangle from this. That's a technical word. Instead of just a hydrogen. I'm just gonna quit trying to get colors. You could have oxygen and a hydrogen, which is hydroxyl group, or you could have, I'll put it at this end. This is gonna look weird. Could have double bond to an oxygen and over here, OH. That's a carboxyl group, which is the standard part of a biologically important molecule that provides acidity. So if you think about citric acid, it's not this exactly, but that's what makes the acid acid. You should have noticed the phrase amino acids in your reading. Amino acids are important because when you link those together, you're building a protein. That's really where we're going here. Okay. So let's see, what did I,
You draw the red.
but the carboxyl group was blue. Okay, carbon is, because we all know coal and graphite, carbon's going to be black. And now, if we do that, now we have that carboxyl group. And on the other side, here's a nitrogen carbon, coal, oxygen, gas, hydrogen, gas, nitrogen, another gas. But when you start linking them together like that, you get all kinds of interesting consequences. Okay, that's a mean group. Mean group over here, that assemblage, carboxyl group. Over here, basic, like the opposite of an acid, amino acid. But what's missing? Some of you know. Pardon me? Ribose, well, if we were talking about RNA or DNA, yeah, but just in terms of the organic chemistry. Hint, how many bonds does carbon typically make? Four. What's missing? Yeah, here. Okay, well, let's. Many colors. Okay. There's a hydrogen. Okay. Hydrogen is kind of a space filler, but it's very, very important. It's very busy in biology. Some other. Red.
It's on the floor.
Oh, is this the sort of thing? We're all familiar with the term X as in kind of a placeholder. It could be anything. In this world, that's what R does. So this is a placeholder. If it happened to be a hydrogen, it would be the amino acid glycine, which is the simplest one. But typically, it's much more involved. Maybe it could be a lot of stuff. So there are 20 different amino acids, and here is where the difference is. All amino acids look like that. It's just whatever that R group is, then you can say, oh, that's cytosine, that's ribosystine, that's phenylalanine, so forth and so on. Any questions so far? I think I'm kind of done with what I tend to do with some basic biochemistry, he whispered. I did find a shirt that has the Krebs cycle on it, by the way. It's really worn out too, and I had to choose one, so I picked this one. Okay. Now, when you're trying to figure out scientifically how to account for life, first we got to go back and think about, well, what do you mean by life? That's what we did last week. And we came up with structure and function at the organismal level, at the cellular level. Remember, the cell is the smallest living entity, right? It has all the bits and pieces, so it can do all those metabolic things, including reproduction and metabolism. Okay. How can we account for that kind of complexity? You kind of know what's been done because you've read those two papers. And what I'm planning to do is to kind of go through them. David, do you have any sense of how many folks are at Zoom land tonight?
We got at least 3.
Okay. So those papers are not scripture, but it's going to feel a little bit like a small group because we're going to not go through line by line. We kind of follow along together, and I'll point out things along the way to see if we can make our way into and out the other end of this. So how can we describe the origin of life in terms of science? Any questions at this point before I get going with that? Okay, let me check my notes. See if I said everything I can do up to this point. Yeah, okay. This quest could be referred to as an historical science. We can't go back and see what happened, but we can, knowing what we do about basic chemistry, put together a set of hypotheses or theories or ideas. You know, there are all these labels. Basically, science, scientists goes, well, that's interesting, whatever it might be, a new kind of toad or whatever. say, well, maybe it's this, maybe it's that. And then you test those maybes, those you could call as hypotheses, I suppose. And you do that enough times in enough different ways, you kind of feel like, yeah, that's not a toad, that's a frog. Okay, fine. And then you continue on. Okay. So folks said, let's think about how we could account for non-living chemicals somehow achieving the organization that's now at least cellular, and we say, well, that thing's living. Now the chemicals really aren't alive. It's the cell and beyond that exhibits those properties of living things. One more, having peeked at my notes from last time, one more reminder where we've been and where we're going. Oh, I don't know, I used to write that much. Everybody hide it there, I can read it. So, wrong red, well. There. Got my own markers because I want to be sure that they were functioning. Okay, so you've seen this last week and now you're seeing it tonight. DNA arrow, RNA arrow, protein. And I suggested to you that this can represent the flow of genetic information. An arrow that I didn't put in last time is this one. DNA is a molecule that has those bases. I mentioned this briefly last time, and a structure, which I won't go through, but I could to some extent, which has information in the form of 4 different letters, but words that are hundreds, if not hundreds of thousands of letters long. Okay. DNA can code for its replication. You can start with one piece of DNA. We do this all the time. Bacteria do it all the time. And make a complete copy of that DNA. So that's what that arrow is for, is information flow from DNA, and now you got 2 molecules of the same, and now you got 4, and you get the picture. This arrow represents RNA synthesis directed by DNA, in that some sequence of the DNA, if the DNA stretches from here to Lake Susan, then maybe you're looking at a piece of the DNA from, I don't know, that fish right there over to David. And now we're going to copy, we're going to make an RNA copy of that short stretch. This is all just very beginning, sorry. So we got an RNA copy of just some of the information here. That RNA copy, if it happens to be, now those are the 2 for next time, so grab those. Now, go ahead, Dan, grab those. You should have the others, I think.
Yeah.
But if you didn't bring them with you, then you can take a couple to,
I just came from fish. So
it's the word priority, never mind. Okay, where we're going, remember we talked about enzymes last time, biological catalysts, and how for the most part, actually almost everything I say in here can be, for the most part, insofar as we know now, but that gets messy after a while. RNA, these short little pieces that serve this role of directing the synthesis of protein, will complex with organelles called ribosomes. You saw that in at least one of your papers, but probably didn't even. A ribosome is this little machine that is able to take this sequence of bases in an RNA molecule, a messenger RNA molecule, and assemble a protein, a sequence of amino acids where the sequence of amino acids in this protein, and you can think of this guy, or just to make it a little bit more realistic, 3-dimensional space, we'll just bend it around here a bit. As many times as I've done this, you think I can just do it automatically, but there, before I get confused. Okay, and maybe this one, this end of the polypeptide. It's another thing you can call this thing until you decide it's big enough to be a protein. There, okay. Where each one of these The black and the white, just different one, but so we got, I don't know, what do we got? I don't know, 26 or something, I'm not sure, amino acids here. There's a very small protein. This sequence of amino acids, threonine, histidine, cysteine. Been a long time. I know all the structures of those ones. Okay. Make this 150, 200 long. What are we talking about here? Is it a hormone? Could be insulin. Is it a structural component of a living thing? Collagen? Could be keratin. What's keratin make? What's keratin make? Look around your cells. Nails? Hair? Epidermis, the skin. And now there's a layer underneath at the junction of the epithelium and the, man, can't even think, what's the dermis? Thank you, dermis and epidermis. Why? My excuse is I haven't done it for 11 years, right? Okay. Down there, cells are dividing, cells are getting pushed up. Those epidermal cells are getting flattened out and drying out. And you know them as dandruff. And the skin is continually renewing itself, pushing out this constantly renewing layer of dead cells, but they have a function. Well, keratin is, there, that's keratin. Like, when the worms, the proteins can be between 50, let's say, and many hundreds, sometimes thousands of amino acids long, the diversity, the possibility for different structures with different functions, that's different. And when you made it longer. Okay. And remember my little fun and games with Dan, who was willing to show up, and Lydia, who was so embarrassed that she hasn't shown up. Remember? And I was the enzyme, and I had my active sites, and they stuck here, and the consequence of them sticking was induced conformational, death. Clunk. And now that hat, which was supposed to be a phosphate group, got transferred from the one to the other. A way to think about that is, remember when they were wandering around the room up here? So, well, one way to make this thing go faster is to increase the temperature. And, then what's your typical stereotype for doing something again, like take a test tube, put some stuff in it, heat it up. Or cooking. Heat makes molecules vibrate and move faster, and stuff happens. But you can't heat up a living thing over the Bunsen burner. Well, you can, but it's not very helpful. So the way that living things manage to not only allow for chemistry to happen at a rate fast enough to support the processes of life, just think metabolism, but it also allows to select. If you've got a starting material, we could call it a substrate if we're talking about enzymes. You got some stuff, and you're going to change it to some other kind of stuff. And it could be small molecules like, I don't know what they got on here. Anyway. Let's suppose there are 100 different processes, reactions that could occur. But only one is the one you want to have happen right now. Just think about the difference between a nerve cell and a blood cell. Pick your type of blood cell. How are they different? In essence, different parts of the DNA are transcribed into messenger RNAs for different proteins. If you're making the mRNAs, messenger RNAs for bone cells, you're going to make bone proteins, etc. Oh, yeah. So you can select from all the different possible processes that are energetically feasible, but you got to get the molecules close enough, long enough, in the right orientation for the reaction to occur. And that's what enzymes do. That's how enzymes, again, this is not quite it, make things go faster. It'll do. Okay, so there's a review of that, because again, if you remember the reading from today, there was a fair amount of talk about RNA and protein and enzymes. And so now I'm trying to warm you up as we go through the papers with that background from last time and this time. So let's start with that after any questions to this point. There is a method to the madness here. I'm building from week to week. Okay. Yeah. Okay. And you notice the issue of the new scientists. The cover. The spark that started life. Okay. Along with, do we need a fourth law of thermodynamics? Yeah. And decoding the chaos inside black holes. Even though this is legitimate, the editors just seem to find a need to, jazz stuff up, make it sexy. So, okay. Unexplained origins. If one puzzled over how life began now, and then sinister protein. I mean, come on, sinister protein. Just trying a little bit of a hook there. Okay. Now, the first, I think, 3 paragraphs are an overview both thematically and historically in this quest by scientists to find acceptable, reasonable descriptions of how life might have begun, in the sense of how we could move from organic chemistry and some inorganic chemistry to cellular structure. That's life. Okay. So Lloyd, what's that first sentence about?
Schrodinger, who was a physicist, but must have dabbled in this stuff.
With his famous book, What is Life, from the 40s. Yep. I kowtow to Lloyd, being his training in physics, because I'll acknowledge physics is real science. Chemistry is kind of sloppy and stinky, and biology is so squishy and wiggly. Who knows what that is? Okay. You can see no good definition. Now, we've been through this, haven't we? In last week's opening. The question of how chemistry morphed into biology billions of years ago, well, that's what we're going to think about tonight. All right. Okay, down in the middle of the, third paragraph, the second paragraph is just one sentence. The last universal common ancestor, or LUCA, which emerged around 400 billion years ago. Okay, now, a lot of work has gone into arriving at that kind of statement, which I'm not ready to go through tonight, but suffice it to say that as one studies genetics and biochemistry, it becomes pretty evident that all life forms have a common origin. Again, speaking in terms of science. And so that's been given the name of the last universal common ancestor. And the timing has to do with both the kinds of physical dating procedures that you heard of. Carbon dating is one that occurred, but that's only good back to 50,000 years ago or so. But a lot of other ways in which one can assess when or how long ago something happened. Okay, so I'm not going to deal with that question unless we have to, but okay. Okay, so now the fourth paragraph. Which came first, DNA, yeah, RNA or protein? And you've read the 2 papers, I trust. Therefore, what I'm going to say now should be, oh yeah, that sounds familiar. Oh yeah, that sounds familiar. If you think about the story I've told thus far, you kind of focus on proteins, because proteins run the show for the most part. Because they're the enzymes, and they're also structural proteins as well, but they're just enzymes. Except that, how do proteins reproduce themselves? And how can proteins carry and transmit, if you will, genetic information? The other choice, and if you recall from reading these 2, is that they each come to the other conclusion. The other possibility is RNA. Why not DNA? It's too big and clumsy. There are many reasons why DNA it's too big a thing, simply put, to figure out how that could have come together literally through just chemistry. Whereas RNA can be quite short, just a few nucleotides along these bases, linked to a phosphate group, linked to a ribose or deoxyribose, and strung together. Okay. So it's either RNA or proteins, those 2 categories that have been the suspects for, golly, 70 years, at least. Well, now we're going to go through and you're going to see arguments for the one and arguments for the other. And remember, this is recent stuff. These are both from just last December. So part of what you're seeing is the process of science, or as I would tell my students and capitalize it on the board, science in action. I gave them plenty of stuff to laugh at me, so they can make fun of me, and that's fine. So science in action, actually studying whatever the process is to figure out what it is and how it works. That's the kind of stuff you do with science. Okay. Okay, now down at the very bottom of this first column, prions are weird proteins. Now you've heard, I trust, of mad cow disease. You also may have heard of, come on, knock up, human disease. Brain death. You got prions in the head. Wasting disease in elk and deer. Same kind of thing goes on in humans in actually two different ways. Wait a minute, why are we talking about disease? Money? What? If you want to do scientific research, it takes money. How do you get the money? Well, you couch your interest in terms of diseases. That's a little cynical and crass, but take too far from the truth. Like when I was doing my research for my PhD and then postdoctoral research, I was funded basically by cancer research funds. Fine. But really what I was doing was trying to develop a little piece of understanding of some basic biology, which was directly pertinent to the process of cancer. But, okay. So as it should seem familiar to you from reading this, prions are these proteins that cause various disease processes that are neurodegenerative. Back to these guys. Proteins, in order to be functional, these chains of amino acids have to be in the right three-dimensional conformation. Everybody's happy with it. They haven't taken out of this position in 15 years. Come on, you can do it. There we go. Okay. So let's suppose, that'll do. Let's suppose that this is not a prion protein. It's the normal, not pathogenic version of the same protein. Now, when people started studying prion diseases, they came to realize when they had some genetic handle on it, oh my goodness, this same protein exists in every from yeast on up, and in this paper, I think they said even bacteria, which I didn't realize. So it's a protein that's been around for a long time. And some specific functions have been ascribed to this particular kind of protein. But if this is that wild type, not harmful form, if you were to just change it a little bit, there, like that. I'll use this to represent the form of this protein that causes these neurodegenerative diseases. And the way that it works, this is insidious. This is amazing. Stanley Prusiner, who was mentioned, got the Nobel Prize, likely, or rightfully so, because what he figured out was, like viruses, which are a bit of nucleic acid, usually wrapped up in some protein. So genes, RNA molecule, DNA molecule, depending on the kind of virus. This is just protein. There's no RNA, there's no DNA. But once this form is in, well, these mammals, I'm not sure if there are prion diseases in other classes of vertebrates, but so deer and so forth, humans. Another molecule, a molecule of the wild type, the non-pathogenic version, can come along and have its three-dimensional configuration altered to this, and they stick together. And again and again and again and again and again. It was likened to a stack of plates, I think, in one of the papers. The problem with that is that when this thing gets big enough, it starts gumming up the works. And that's the essence of the pathophysiology of prion disease. What does that have to do? It has to do with our topic at hand in terms of understanding the importance, the role of the right and the wrong configuration of proteins. Let me go to the paper now, but first, any questions thus far? Okay. Okay, so the first paragraph on the right-hand column, life requires metabolism. We've visited that idea already. And then in the middle of that, but enduring isn't enough. Life must also reproduce. Okay, talked about that a little bit, and you can read what's there. Then in the next paragraph, reproduction simply meant molecules that accompany themselves. So at this level of organization of matter, we say reproduction, we're thinking about, well, how can one molecule manage to create another one of itself? How can the molecules reproduce? So part of the hypothesis or the group of hypotheses is, well, if we're looking to move from just water molecules, carbon dioxide, ammonia, and so forth, to these larger molecules and then more of them, well, clearly then they have to do that. You know, there's no living entity there already, right? Okay, so how do we account for that? Okay, so you can see midway through that, what is it? Well, the next to the last paragraph, starting on the right-hand column, there would be no life if metabolizing replicating molecules hadn't appeared. How this happened is the central question for scientists working on abiogenesis, the emergence of life from inanimate chemicals. So there's the question right there. How can chemicals develop enough complexity to both provide for their own replication and the necessary machinery to do this thing we're calling metabolism? And neither paper really talked about cells that much, but also the structures of the many different machines, we call them organelles and other things, in a cell. So there's a lot more, but get the idea. Okay, and so there we have now at the bottom, the end of this first page, the classic piece of research done in the 50s at the University of Chicago. Let's see. Miller then convinced the reluctant Urey to let him test his hypothesis that life started in the ponds rich in salts, atmosphere, hydrogen, ammonia, methane, electricity for energy, and maybe other aspects. This is not atypical. Okay, PhD student says, you know, and the guy that's providing the funds, you know, the guy that wrote the proposals that turned into grants, and there's the money. He's got his own thing he's trying to get done, and you're in the lab there, ostensibly helping him with that. Yeah, and that's what his research funds buys. It buys materials, some infrastructure, and cheap labor. PhD students and postdoctoral students. Okay. And yeah, but I think I want to do my own thing, boss. And the boss says, well, okay, you kind of do it over there on the side. Don't make a big deal about it. And son of a gun, after a couple of weeks of this, it was a round bottom flask and a recirculating thing. Not DNA, not RNA, but at least some amino acids and some other chemicals were found in the mix. Well, look at that. Interesting. Starting along on that path. And then in the paragraph that starts on the next page, so we're on this page now. Since then, arranged environments and so forth. Okay, where did life start? Well, the shallow pond was one idea. Vents deep in the ocean is another idea, where you have very hot water that's been heated by geothermal activity bubbling up with some unique chemistry going on just because of what chemicals are there. That's another idea. These are all kind of interrelated, really. Another one is, well, back to catalysis, enzymes, a surface, a clay surface, because a clay surface has some neat little stereochemical pockets that provide neat little environments, almost like the active site in an enzyme. So that if you have one molecule here of, I don't know, methane, and well, one molecule over here of carbon dioxide, whatever, ammonia, stick right next door, just like I was doing last week. That's how something, well, a kind of clay surface, or now many other, a surface can provide an environment where catalysis happens. The reaction occurs because effectively you took this molecule from here, that molecule from there, and they're just bouncing around, and this guy gets stuck, bouncing around, they get stuck. And that concentration is effectively really high because they're right next to each other instead of being out here, just like I did last week with my volunteers. Okay, now continuing on, let's see. Oh, yeah. And the idea of having an environment where at least one of the properties, typically temperature, is called upon. Reactions that happen at a higher temperature, we've thought about that, can then be shut down by lowering the temperature. That also means that if you made some products, had some chemistry happen at the higher temperature, and then the temperature gets lowered, the chemistry stops. And any of those products you made stick around. That's another piece of the challenge. How can we go from these itty bitty molecules to bigger and bigger ones? And part of what has been proposed is exactly that. Some means of maintaining the products once they are made. You saw the word quenching, I think, in both papers, which refers to, in part, that idea. Okay, when life on Earth. Any questions? Yes, sir.
That'd be the day and night cycle, but change in temperature.
Thank you. I didn't finish the story. Yeah, so what can account for or provide such a fluctuation? It could be day and night. It could be tides, moving stuff from one place to another, or at least bringing in cooler and hotter. A variety of different phenomena could be suggested as providing for a fluctuation. Maybe it's pH. Maybe you have two streams that run together. Maybe it's in the bottom of the ocean, but streams of, and they're different pHs where they run side by side, or better yet, go cross current this way. So there are a lot of ways. Just thinking about physical phenomena that we already have known, have identified as real. Call on that kind of sidebar. Have you heard of PCR? Heard at least one, maybe. Okay. Anybody know what it is? It's used a lot. Pardon me?
Polymerase chain reaction.
Yes. What is the polymerase chain reaction? Thank you for volunteering.
It opens the DNA and
You're on the right track. Yeah, internal medicine doesn't really require this. Yeah. So here's the deal.
Biochemistry a long time ago.
I did too. But anyway, I have not done any kind of medicine. I bring this up as an example. Polymerase chain reaction, all those TV shows or news reports, they talk about DNA and trying to determine if a particular crime scene sample matches the suspect. And what they're doing is some technology by which that can be achieved. Compare the DNA sequence of the one to the other. Part of that process, not the whole thing, but part of that is here we've got a piece of DNA, but we know that this sequence right here is a useful sequence. There are a bunch of reasons that tend to be technical. This is a sequence that is useful for comparing DNA from this person, that person, the other person. Okay, how do we do that? Gather the DNA from the two samples, so crime scene and a database probably at this point. and take that crime scene sample, you already know you're looking for this particular sequence, which varies from person to person. Okay? It could be a gene for a protein. It could be. And you have already determined that if you now take that, melt, that's one word, the two strands of DNA, separate them, and then you can drop in some short little pieces of DNA that you've synthesized chemically that match the sequences here and here. Drop them in, and then provide the necessary, I wasn't planning on doing this, provide the necessary substrates and enzymes. And now you'll copy, and now you have two strands where you had one, except that if that DNA strand actually went from the wall to Lake Susan, we've only copied this stretch. Do it again, and pretty soon you've got millions and millions of copies of just this part of the DNA from the crime scene sample, and then compare that to the sequence data that's already been done on various. Now, what does that have to do with what we're just talking about? They said the way that the part of the process of PCR, polymerase chain reaction, is that the process of synthesis goes on at ambient temperature. So think body temperature, something like that. Okay. But to do it again, you need to separate those strands. Well, if you heat the DNA up enough, the two strands will come apart. It also stops the reaction. It also kills the enzyme. So people figured out, oh, we got to isolate enzymes from bacteria that grow in hot springs in Yellowstone. Seriously. And so the mix gets put into these little tubes. I've done it. And just program it. And it's just a cycle of temperature goes up, temperature goes down, temperature goes up, temperature goes down. And over a period of a few hours, if everything's set up properly and it's working, have millions of copies of that stretch of DNA that's this long from some piece that's that long. And you now compare them and say, nah. Eventually, though, perhaps, ding, 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 they match adequately. Okay, so there's an actual current process that's being done that involves a cycling of temperature in order to move from a small number of big molecules to a whole lot of them. Okay, that wasn't even planned. I should press on. You are being very alert, or at least you're putting on a good show. Thank you. Okay. Okay, when life emerged, hot springs, thermal opening, and so forth. That's and you've read that. Let's see. Okay, now at the end of that paragraph, although Miller's experiment suggested life began with proteins, of course, he kind of rigged it that way. Just put in some starting material, methane and so forth, that seemed like a reasonable place to start. You'd be hard pressed to get a nucleic acid or nucleic acid precursor that way. But anyhow, now the leading idea in this paper is that RNA came first. And that goes back to the 1960s. Francis Crick. Lloyd, what was Francis Crick's profession, training, and expertise?
Physics?
Yes. Yeah. Watson and Crick, the two guys that figured out the structure of DNA, which really allowed this whole story to start to be told. Watson got his doctorate in biology at the University of Chicago as an ornithologist, a bird watcher. Yeah. Crick, and he was a fresh off of his degree that he traveled over to England. Crick was, I don't know, 38 or something. So he'd been around for a while, had done radar stuff during the Second World War. And the two of them did this amazing stuff. They didn't do any experiments. They stole Rosalind Franklin's X-ray diffraction image that they interpreted correctly as a double helix and story. Anyway. So now here we are 10 years later or so with Crick and others trying to ponder the problem of how do we account for the, yeah, that is an hour back. Okay. How to provide a reasonable explanation for life arising from inanimate matter. How about RNA? Okay, and you can see in the next paragraph, the last full paragraph, these organic components formed spontaneously and were subsequently linked together to make RNA. Okay, and RNA. So RNA is a linear molecule made up of nucleotide phosphates strung together. So now I'm going to say, okay, that's mRNA. That's RNA. Well, it's protein there for a while. I'm going to call it RNA. And its form, its shape, can definitely affect its function. And remember ribosomes? We talked about those very briefly. These machines inside cells where RNA directs the assembly of amino acids according to that sequence, synthesize a protein. Ribosomes, and I think you saw this in the reading, ribosomes are these complicated, blocky, chunky things where imagine now that we have a couple of RNA molecules, so make this 60 times bigger anyway. And now start lumping a bunch of this is no longer prion protein, it's just a protein. Actually, it's a histone. But okay, start lumping this around, and you get enough of that. That's what a ribosome instruction. It turns out, that the enzymatic activity of ribosomes is actually part of the RNA molecule, or two of the RNA molecules, not the proteins, which is counter to what everybody thought. And there's some other examples of RNA molecules that do have enzymatic activity. And you saw the word ribosome, not ribosome, ribozyme, R-I-B-O-zyme, as the label for RNA molecule with enzymatic activity. See where was I? Because that never had to deal with sound coming from electronic devices. Did this when I was teaching. Okay, back here. Okay, so they're in the middle of that paragraph. So right down here, there's where you get ribozyme. And there would no metabolism without enzymes. And another argument in favor of RNA is, hey, RNA can, in fact, does contain genetic information, base sequence order, just like in DNA, but in RNA and the chemical differences between the two. Okay, okay. Okay, and so now we're at I think your paper probably has a prettier in color picture than mine. That's of hot spring, or that's in solid. Okay. Like DNA replication, this requires enzymes called polymerases. An enzyme that catalyzes the joining together, the polymerization. And it turns out that some RNA molecules can be demonstrated to exhibit that kind of activity. Okay, so the next paragraph, well, can we do something with this? And in essence, what was done was to, in a way upsetting, of course, in tubes and so forth, run a series of 11 sequential processes of messing around with the fidelity of the replication so that mistakes were introduced, mutations, but under certain selective conditions, pushing the system towards a particular kind of activity. And after 11 cycles of doing that, yeah, hey, that actually worked out. In the next paragraph, the RNA world hypothesis has some flaws, however. Well, of course it does, because this is the paper that pushes for proteins. And RNA molecules are very unstable in water. Yeah, DNA is pretty darn stable. RNA is not. If you're going to do RNA, I'm sure they figured it out. When I was a kid, doing DNA was easy, but doing RNA was a challenge. Because yeah, it's just very unstable. Okay, working in the lab, that is. Okay, one of those experiments, middle column. Okay, Simpson, okay, fine. Okay, so now we're to the RNA problem. The same cannot be said of RNA. So having said, hey, look, proteins really fit, really work, RNA has some problems. Spontaneous formation of purine and permanent bases and ribones. Well, okay, remember when I drew an amino acid? It was a carboxyl group here, an amino group here, one carbon in the middle, and a hydrogen and some dangling. Okay, that's the subunit. That's what you string together to make proteins. I didn't get ready for this at all, so it's going to be not quite right. I'll just make it really, really simple. Okay, there's a nitrogen there. Again, I used to know all these structures and oh, wait a minute, what are you doing? 7th or 8th grade geometry class here. Here. Okay, each one of those corners represents chemical bonds coming together. Most of them are carbons. There's a few nitrogens. I marked maybe the ones that are nitrogens. Without all the other stuff. Okay, so I just draw an amino acid look like. So these are the bases, the nitrogenous bases that are part of what makes up DNA and RNA. A little bit more complicated, for sure. Plus, then you also need ribose, which is a sugar, 5-carbon sugar, and phosphate groups. Those are not so complicated. So that's what the paper is getting at. Let's see the paper. Okay. Building an RNA world from scratch requires the spontaneous formation of purines and pyrimides. Yeah, that's no small hump to get over. Okay, never achieved in the laboratory, and okay. There's another advantage to the protein first idea. Okay, nearly all enzymes are proteins. Unclear how a protein-only life form could have reproduced, but, and now we get back to the prion story. Bringing in to play, oh, here's a protein that can catalyze the synthesis of itself. Yeah, it's starting with a protein that's kind of the same, but still, there's a process. Yeah, okay. And so the second paragraph that starts on that right-hand column, Stanley Fruser demonstrates that prions do not have nucleic acid. Wow. And then next paragraph, hey, prions or the prion-like proteins are throughout life forms. And then the conversation about folding. And let's see, I've pretty much done all of that. Well, the final paragraph that starts on this next to last page. Prions are a type of intrinsically disordered protein, which don't fold spontaneous into stable shapes. There's a whole lot there that I could talk about that. They constantly fold and unfold into many thousands of unstable shapes. This is partly how all proteins work, but when a protein is synthesized, think of amino acid to amino acid to amino acid, it starts getting long and floppy. And it will fold up on itself, and it might be directed by other proteins, folding or. So just sticking the amino acids together in order, that's a big job, but now the thing has to fold up properly to function. We're back to structure and function, folks. Okay. And now we have, the miscoded, misshapen form of this protein, the prion protein, that now can serve to lock down all of the non-pathogenic proteins into the pathogenic three-dimensional structure. Let's see, and then, okay. And so in the middle column of this last page of this paper, being prion-like, they would also have been able to replicate, enriching them, okay. And that still leaves one problem in the business about how do they stick around. And now on the upper right, the first paragraph, the ribosome results, okay, talk to you about that. And basically talking about the interaction of proteins and RNA, which is a big theme at this level of biology. Okay, proteins may exist doing their thing without RNA molecules, but RNA molecules, lots of, I've told you about mRNA, there are a lot of other RNA molecules, kinds of RNA molecules, and they most always have specific proteins stuck onto them. And so they function not as naked RNA molecules, but as, here's one for you, a ribonucleoprotein particle, blah, 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 which you could consider a ribosome as well, I suppose. Okay. Okay. I'm going to stop from this one. Please ask questions, things I skipped over or things that I said that, yeah, whatever. What?
I underlined and made note of a few words in here that strike me.
Okay.
In the middle column on this last page,
Okay.
about 6 rows down, it says, by chance, the
Yep.
sequence of some allowed them to form highly resistant fibrils, probably unlike. And then later in that, just before the fibro survival, the second line above that says, may have built up.
Mhm.
And then over in the right-hand column, in the second paragraph, about a third of the way down, protein worlds emerge spontaneously on the young Earth. Those words are key words to me.
Okay, I can respond, or maybe you should explain why it is that those particular words caught your attention.
Well, because chance is you have to have faith in something, so you have faith in chance, which is not a good chance. And anytime I see may have or might have or possibly, it always makes me think, okay, you really don't know what you're talking about, but it may be. And spontaneity is just some without any good explanation.
Now, obviously, coming from a Christian background, as our resident physicist does, he's catching on to things that sound like people are trying to avoid saying God. And that may well be the case, but we're not. Think of that diagram. We're down in the one corner. God is not an answer. It's not a viable answer if we're doing
Correct.
science. So we are left with these sorts of things. Now, in a couple of places, in one or both papers, it says about, well, this is a 1 in a billion shot, right? Say goodbye. Chemistry. Let's go to microbiology. Let's go to bacteria growing in cultures.
What we call those hedging words.
Oh, yeah.
That's what they say on the news. And they're trying to put a slant on it. Could have done this. And in the finance world, they say it might have done this, or this is what we think it might do. And then you think that's the gospel. Scientists use it a lot.
Well, yeah, it depends on who says it.
Yeah.
If the accountant says it, it means nothing, but.
This, I'm gonna deal with some of these in a more direct manner, I think. But I think we all acknowledge that as human beings, we are limited in our understanding of anything. And so honesty has that ambiguity expressed. The people that tell you, well, they're obvious,
This book.
This is a dry erase mark, I mean, who wants to argue with that? That was trivial, but you understand. Okay, let's go back to something here. This is not a trick question. Yeah. What's that? Sorry.
Well, if it's binary.
It's all binary.
I never learned binary, and I pretended I could try to learn base 6 or base 8. Who am I kidding? So, I'm base 10. In fact, this is about base 10. It's 100. Okay. What's that?
10, just 10 squared.
10 squared, which is 100. For many of you, it's like, yeah, I've seen this before. What's the exponent?
3.
3. 10 times itself 3 times. About. 6. Yeah, that's a 6. 1,000. Million, boy. You know, I find that embarrassing. Let's try that again. My wife thankfully won't bring this up afterwards. You guys are stuck having to make sense of my errors.
Much better.
Thank you, ma'am. In our household, the proper response can be yes, dear, but given that she's a retired colonel, it's yes, ma'am.
Finally learned.
Well, I've had, what, 50 years, almost 53 years to get it. Okay. Okay. So if you were to use the thinking of 10 to whatever power, it's really, really useful when you're dealing with big or small numbers. Oh, well, you know, that's a 1 in a million shot. That's just a popular way of saying, ain't gonna happen. Well, what it really means is, it happens at a given rate, very small rate, but you may have a hard time finding it. Okay. Now, when you're growing bacteria in culture, so think of a tube or think of a flask with kind of a beige colored solution in it, and you inoculate it and let it slosh around for a day or 3. It gets cloudy, the bacteria are growing. And the density at which those can grow to is on the order of, depending on things, 10 to the 6th cells per milliliter. Oh, cubic centimeter? I don't know, that's medicine yet. Milliliter for the scientists in the crowd. I don't know, that much. Okay. 10 to the 6th per milliliter up to maybe, well, 10 to the 8th for sure. 10 to the 8th cells per milliliter. For a given genetic locus, that's a place in the DNA or RNA, depending what we're talking about. For a given genetic locus, depending on details, rate of mutation so rate of event per replication cycle. So 1 cell goes to 2, or 100 goes to 200, or 2 million goes to 2 million. Okay. Mutation rate. Boy. I'm going to put this fellow in bed. Mutation rate. If it's a system that has not good proof reading, seriously, is typically taken to be on the order of 1 in 10 to the 4th, which, by the way, is 10 to the minus 4. I tell you, big and little, you don't have to figure out. You know, when you're filling out a form and they gave you your driver's license, whatever it is, and it's got 5 zeros in a row. And I don't know about you, but I got to use a physical thing to block it and count it 6 times to make sure I got the right number of zeros. Right? Okay. 1 in 10 to the 4, which is 1 in 10,000. 2, maybe 1 in 10 to the 6, which is to say, there's your 1 in a million shot. Per milliliter. In a tube, you typically have 10, 12 milliliters. In a flask, let's make it easy. Let's call it, well, not easy, 200. In this flask, it's been sloshing around for 3 days. And let's suppose it only grew to 1 million cells per milliliter. And we've got 200 milliliters and at a mutation rate of 1 in 10 to the 6th, which is lower than this by a factor of 100. 200 milliliters means that we have 200 1 in a million shots present. A 1 in a million shot is certainty in certain situations like this. It is certainty. The trick is to find it, and there are all kinds of tricks that you can teach students to do that. That we're not going to go into tonight. So when I read something like, oh, well, it's a 1 in a billion shot, I can't help but kind of shake my head and laugh. We're supposed to be impressed and awed, and okay, that means it doesn't exist, can never happen. In my mind, that means it's a guarantee it will. Eventually, this is, over the course of, let's say, 3 days, 72 hours. But given enough time and the circumstances for that mutation, that change to then be stabilized, we're back to thinking about the molecule, stable and get more of it and so forth. So a 1 in a billion shot, my way of thinking, is also certainty. It's really, really small, and you may have a really hard time finding it, depending on what it is, but it is certainty. Okay, so I have a hard time getting to, go ahead.
It's a little random. Decided of Luca.
Yeah.
Never heard of this before. It's described as an organism, so does that mean it is living?
No. No, what has been done is to examine all this biochemistry and molecular biology and say, well, from what we know about living things, we can see how this arose from that and so forth and so on. Go back a certain distance looking at extant living things. And then say, okay, now we can look at the geological record and find evidence for life forms. You know that how are we doing here? Oh, okay, we're about out of time, right? Yes? Okay, I'm about an hour off, and I haven't even gotten the second paper. I'm sorry, what was your question? I'm just trying to.
So it was not a living organism?
Following that information, I was going to talk about different isotopes, but let's not do that. It's nothing we've ever seen. Okay, but again, if you accrue all of this data and look for the most reasonable interpretation, you find that this arose from that, arose from this, arose from that. And here we are up here, and now we're looking backwards with what we have at hand, which is an understanding of molecular biology, biochemistry, and genetics, and what we can learn from the geological record, not in terms of how old, well, that's part, but the presence of certain kinds of chemicals. Hard to explain except by saying something was altered. That's a biological product. And then that's an area of some discussion as to what counts. And so Luca is that label for, well, it sure looks like everything came from one common ancestor. And so the last universal common ancestor is that. So it's nothing that we're ever going to see. It's part of this historic science where we're trying to tell a reasonable, comprehensive story based on the evidence that we have.
I understand it. I see it.
Yeah.
It's a common ancestor, then it must have reproduced.
Yep.
And it's reproducing, isn't it living? Isn't it a living thing?
At that time.
Right.
Yeah. Oh, yeah. Sorry. This is the label for the first time that matter became organized enough to display the properties of living things. Therefore, we can call it the first living thing.
And we call it the first common one. So the implication is that there was, or there just might have been other organisms that did not survive,
Didn't make it,
didn't make it.
or were there, but they didn't really do such a great job, and so they got pushed aside.
And presumably they're gone.
Yeah.
They didn't go anywhere.
Yeah, they're gone.
My other question is unrelated, but my DNA is different than my mother's and my father's. It's similar, but it's different, right? Do I remember that RNA is not like that? Is my RNA different than my mother? Father?
Your parents and you have within the DNA a gene that codes for a particular enzyme that determines whether you're type A, B, or AB or O blood. Okay. And we know for that example that, you know, if mom's O and dad's O, kind of hope you're O, right? Et cetera. So there's that kind of continuity, that kind of, yeah, they're the same. But even with that, the gene, let's assume it's O, the gene from mom, O, the gene from dad, O, are probably not identical. There are differences in the sequence, but differences that don't impact the successful production of that enzyme that accounts for type O. Is that helping any?
I'm trying to remember this story about the universal Eve. A few years ago, this was, in my mind, I was thinking that they were able to trace common ancestor of us all, the Eve. And they were able to do this because the RNA didn't change or something. I probably had that wrong, but
Hey there. What was the other paper on? I'm going to assume you read the other paper. It made sense. And go back and read both of these now. And I bet they'll make more sense because let's follow this. In trying to understand something about, I'll say human development, you know, say I'm a way to get trouble. Say development's okay. You can compare the DNA across sequence, across groups of people. Another way is to compare the sequence of RNA from the two, well, there's a third one, the two and specifically one of the RNA molecules from ribosomes, because ribosomes make proteins. They pretty much are this, well, they're very similar across all life forms. That is, if the sequence of an RNA changes, that's not going to happen very often because most times the sequence in an RNA changes. The sequence in the gene that codes for the RNA changes. It means that the ribosome isn't going to work. There's very little room for flop in that sequence. And you can isolate ribosomes very easily and then compare sequences from the ribosomes of this group of people, that group of people, and so forth. And get a sense of, you know, think about broccoli, cauliflower, kohlrabi, various different Asian vegetables. They're officially, at least most of them, all the same species. But clearly they're different, okay? And so when you're looking at life forms and trying to pigeonhole them, you tend to put like with like and so forth. But then are you doing that at what level? Oh, this one's green, that one's yellow, they're different. Or, oh, this sequence of RNA, the sequence of the DNA that codes for the RNA. Doing that, I'm wrestling how much to use where you guys have been very patient. I'll take the simpler one. You pick maternal, which I could go on with. Males have Y chromosomes, females don't. Okay, humans. Let's apply the same approach to the sequence of DNA in the Y chromosome of humans. This is fascinating. When I read this is like 20 years ago. It's like, that's just so cool. Think of the last name of Cohen. Typically spelled C-O-H-E-N. I don't know what it is here. That's a hint. What do you know about the family Cohen? Jewish. And the plural of Cohen is Kohanim. I don't know if that helps any. It's the priestly family.
The Levites.
Huh? This kind of analysis is applied to various groups of people, including that family. And you know what? They clustered into one little group. Really? No, that's not millions of years, but it's tens of thousands of years anyway, or at least 10,000 something. Really? That's just to try to bring this very abstract bunch of stuff I've been yammering about to something that you could identify. Here we got this group of people that will claim socially, culturally, to be a unique group within this larger unique group. And the molecular biology says, yes, you are. That was just too cool. And that's how I'm going to stop, because that's kind of a high point, I think. But please, are there any other? I know you're afraid to ask because I'll just go on for 20 minutes and you're right. But
Could we conclude something about the origin
go ahead, try. You want me to say something?
Well, I have some thoughts, but I value yours more than mine.
Okay, again, the point of this exercise tonight is to show you the manner in which the process of science can be applied to the question of how do we account for the origin of life. One way to think about it is, well, duh, it's easy. Plants do it all the time. Plants photosynthesize. They take CO2 and H2O and make glucose. Yeah, but glucose, complex. Or we eat. Yeah, it came from something that was alive, but still it's dead. And now it becomes part of us. So the point is that clearly taking non-living matter and turning it into a part of a living thing, that's routine, happens all the time. But how do we account for how that first happened, the origin of life, the appearance of the first structure with the necessary details to do all those functions that we associate with life? That's what tonight has been about. And yes, there's a whole lot of, it could be, it might be, but that's the way science is.
Has it ever actually been done in the laboratory? But the Miller's experiment was the earliest one that I've been aware of. Apparently, there were many others, to which they got amino acids.
Yes.
But they didn't get a living organism.
No.
And so far, we can conclude that we haven't been able to do it. And if we did do it, we did it by design.
Yeah, that's more of a philosophical gotcha. You're quite right. What we haven't considered, we just, if you think about organization of matter, atoms, molecules, macro molecules, meaning DNA, RNA, protein, and then on to structures that it used to just be organelles. It's more complex now, of course, but things that you only find in living things, organelles, you find them in cells, of organization of matter that you're not going to find outside of living things. And then wrap it into a balloon, a cell membrane, phospholipid bilayer with embedded proteins. And now you've got the structure that at least all living things share. Was Luca that complex? On the one hand, probably, but it probably wasn't as complex as what modern cells are now. And so we've been knocking around down here at a lower level of organization than cells. So Lloyd, in one sense, we're not even close. But when did that stop scientists from asking entertaining questions? So folks are definitely working on it. Phospholipid vesicles, ever heard that phrase? Probably not. That's part of how the RNA vaccines for COVID. Part of it, it's an mRNA molecule, but the structure of RNA is very unstable. But hardworking, brilliant folks in a very brief period of time figured out, oh, we can chemically modify. They would isolate an RNA molecule that is clearly from COVID, something that would get the immune system to go, oh my goodness, we got to do something. They chemically modified at least one end of the molecule so that it could not come under attack by exonuclein enzymes that will gobble it up from that end. And then they made little tiny bubbles of phospholipid, certain specific phospholipids I won't get into all that. And that's in essence, and there's some other things. That in essence is what the vaccine, the mRNA vaccine, Moderna and Pfizer. Well, that I forget what guy. you want to go home and do something else yet. No, I'm totally enjoying this because you guys are paying attention. I know you're not freshmen in college, but thank you very much for your attention, and we can continue. I'll be here next week. Good to see. I'll do a little bit, Mark, a little bit, just see where it goes. But thank you all.